All right, so today we'll quickly go over some microbiology techniques, which you already know. You have done some practicals on these things in the lab, in the microbiology lab. You did these things, but this time around, you are only going to tune your mind to the industry. You know how you apply this in the industry. The same microbiology techniques this time around in industrial settings. And the first we are looking at is submerged fermentation. So it's one of the ways that uh, industrially there is that production of enzymes, because of course it's the enzymes that you need enzymes to catalyze biochemical reactions industrially in your um, bioprocess in your processes or the bioprocesses. uh, you want to have, when you have some feedstock and all of that, you add some enzymes to it, and these enzymes themselves you do produce them from some microorganisms, and you do that by a process we refer to as a submerged fermentation. So this is like the way you do culturing. But this time around, you are doing this culturing in a liquid medium, not on a solid surface. And if you look at the diagram on the right, the submerged fermentation, you see there is so this is a representation of say the bioprocessor, in which you have the liquid media, in which of course you have some microorganism introduced there, which will be producing some enzymes. And you notice that there are some inlets. So you have an inlet for, say, maybe acid, inlet for base. So this is just a representation. You have an inlet for some fresh medium, which is like the feedstock on which you have, the microorganism feeding. And of course, you have some air, oxygen also in there. Then you have a rotor. I mean, the rotor is responsible for keeping that um, medium homogeneous at all times, because as it rotates, it mixes the media, in which you have this whole reaction mixture, which will be kept homogeneous at all times by the rotation of the rotor. Now, this process is used for the production of different kinds of enzymes, like say the amylases, proteases, lipases, those same enzymes that we talked about. And in the last lecture, we did even talk about, how these enzymes are applied in different industries. And so now we're just looking at how do you apply the microbiology, which you had studied in the industry, to produce, enzymes such as this. So if we have some microorganisms, some bacteria that produces amylases, or maybe some yeast, some fungi, you can grow it in a media such that those substances, because once those microorganisms, because they will be growing in the media, and once they grow, they will be producing enzymes, and those enzymes are proteins which can then be harvested and purified, and then they can then be used in a particular industry, maybe in a food industry, or we even saw the use of it in, say, textile industry, or the use of it in a beverage industry, in the detergent industry. We did see all those things. So you can use such a submerged fermentation process for the production of these proteins. Now another form is just like the common culturing that you do with a solid media in a petri dish in the laboratory. But this time around at an industrial level, such that you do this culturing, at a mass level. So if you have here in the middle, say a bioprocessor in which you have different layers like shelves on which you can have different solid media that are inoculated with the microorganisms, and that's bioprocessor. Yeah, you have optimum conditions set in it, so that you have an inlet for some air, because the microorganism will need some air. Of course, when it's also making use of the air and producing, maybe the enzymes that you need, there will be byproducts as well. And so you have an outlet for air as well. You also will have, even though it's solid state fermentation, not the liquid one which we just saw, still there will be the need of some amount of moisture in a bioprocessor for the microorganisms to produce the enzymes that we are looking for. And so there will be some inlet also for some water supply, except that that is controlled, so that it's just going to be in a form of uh, some vapor, which can keep the bioprocessor environment moist enough for the microorganisms to grow and to produce these enzymes. Then, of course, you also see there's a place for the discharge of the water which you introduce in there to produce this vapor into the system to have an optimum humidity in such a system. Now we use this solid state fermentation for the production of different enzymes such as cellulases, pectinases. I think we saw these cellulases, pectinases particularly being used in the textile industry, in fabric polishing, biopolishing, and we even saw it being used in the food industry, like also the breweries, so that for the increase of yield when you have the breakdown of these indigestible carbohydrates like cellulose and pectin, which will add up to the carbohydrates which are easily broken down by the amylases in order for high yield production of, say, maybe the beers or the beverages which are being produced from carbohydrates. And of course, usually you find that in the biodigesters that these maybe enzymes are produced, you have media which is used a lot of times is made from some agricultural residues, for instance. To produce these things at a large scale. So is that kind of same thing which you have been doing in the lab in a petri dish, but this time around is look at it as it's a large petri dish, and even different multiples of them at different layers, and you have it in an enclosed bioprocessor in which you have the optimal condition of, say, temperature, humidity, pressure, all those parameters that affect enzymatic activity. They are all optimized in there, and then you have the production of these things. Now, these things are not manual per se, so it's not things that you should. I think what I should do is, last year I was giving a lot of videos to your friends to do some kind of a virtual excursion to see actually in reality how these things work. Except this time I'm not having access to some of these videos. Anyway, when I come to Ghana, I will get those videos and then I'll give them to you. Sometimes even maybe when we meet like this, what we do is we watch some of these videos and see how these things are actually done in reality. Because this course is not one of those that should be, how do I say, reading a lot and talking a lot and things like that. But you should actually see these things happening in the industry because these are very practical things that if you're just reading and seeing it on paper, in books, uh, you still not have an enjoyment of it. You will not see really how these things are in action. But when you see it, at least, even if you are not in the industry yourself, but you see it in videos, I mean, you better appreciate it and like it and see how actually the things that you have been doing in the lab are being applied. I mean, in reality, in real life. Right. Then we talk of also batch fermentation. So we are classifying the fermentations in terms of the systems that are used, the types of bioprocesses that are used. Starting from the first biodigesters that have been used, and then we'll continue up to say in modern times what is the most industrially optimal system which is being used. There are just basically three of these things, and the first of them is the batch fermentation. And in batch fermentation, what it means is that you see the growth curve which you have always referred to in microbiology, where you have a lag stage of bacterial growth, exponential stage, stationary stage, and a depth stage. You now apply this industrially, knowing that when you introduce a bacteria to a medium, it takes a certain time for the bacteria, to grow at an exponential level, and then it reaches a point where it's at a stationary level, and you can have the highest amount of the product you are looking for being produced. So you can determine your production time just by knowing this life cycle of the bacteria. So for instance, if you know that the bacteria that you are introducing into the digester, into the processor, is going to grow over, let's say, that exponential the exponential growth period is going to take maybe some 12 hours or 24 hours. And then a stationary phase is also going to take maybe another maybe 48, 72 hours, things like that. You can set your system, your bioprocessor, to cover all this period so that at the beginning of the period of cell death, that is at the end of the stationary state, when you have the highest production of your product, that will be the end of your production time, and that will be the beginning of harvesting of your products, so that you don't have a lot of these cells dying in that mixture and perhaps contaminating the product that you are producing. And what is the batch system? The batch system simply is that it's an enclosed system that when you introduce your feedstock, your bacteria, you close the system. So everything, the bacteria, the feed, you set all the conditions. The system is closed. You set the time, and all you have to do is just to wait for the system to go on, to go through all the cycle from lag stage, exponential stage, stationary phase, until it gets to the last, the the end of the stationary phase when you set the time, and it means at that stage you have end of production before you can now start to harvest. So that's a batch system. It's a system which has been used from the beginning of the introduction of these fermentation processes industrially. That has been the first system that has been used. Of course, it's still being used in many cases in industries. Of course, it's also in the production of enzymes like proteases, amylases, where you do use such a system, the batch fermentation, to do that. And if you look at a system, it's just similar to what we just saw much earlier when we were looking at the submerged fermentation. You have air inlet coming in there. You have the medium where you pass the medium through into the bioprocessor. Same place, you can pass, you will have a pump that is, pumping this medium into the bioprocessor. Then, of course, we saw that there was a rotor, which was, rotating and keeping that mixture homogeneous at all times. That's what you are seeing here as an agitation system. Obviously, it's there has to be some enclosure of the system, to create it's kind of a thermostat to maintain a certain optimum temperature for these microorganisms to grow and produce the product of interest. Of course, there are sensors that will be detecting at every point in time conditions of the reaction mixture, and what are the conditions that these sensors are going to be detecting and registering. The conditions are those we looked at when we were studying the enzymes, looking at things like concentration of, say, the substrate, which will be the media. The media you introduce in there will be containing some specific nutrients like carbon, hydrogen, oxygen, nitrogen. In some cases, there will be some sulfur and there will be some phosphorus. So you have sensors, that record these, the concentration levels of substrates, for instance, and then conditions like temperature inside a reaction mixture should be monitored. pH should be monitored. And these things actually will be showing on the screen once you set up the system and the processing is running. You have these parameters that are being monitored. You will see it on the screen on the monitor, and you have these sensors such that, for instance, if you are having too much of a pressure building up in the bioprocessor, of course there should be a sensor for also sensing pressure. It will quickly give an alarm, maybe some sound or some light beeping, to call your attention so that you can attend to such a problem. So you have such a system that once you feed in everything, you close, you just wait until the process is over. You don't do anything. When the process is over, then you do a harvesting. Then you have what you call the fed batch fermentation. So this is an improvement of the batch system, because this system is going to produce more than the batch system. Why is it going to produce more? Because you see from the term, it says fed batch. So this system is designed in order to prolong that growth phase and increase the yield of the fermentation process. So once the growth phase is increased, it also means that you have stationary phase also going to increase. Because what it means is that if you have a system, so you see what is being depicted on your right-hand side with the curves is that, first of all, you have the red curve, referring to dissolved oxygen tension, which is to say, concentration of oxygen in a system. And then you have, some substrate concentration, which is the dotted violet line. Then you have the feed, feedstock, what you feed into the system for the microorganism to be feeding on. And you notice that, if this was a batch, the red line from the beginning is showing that there's some batch that was ending, and just when it is ending, there is an introduction of a feedstock into the system. And because of the introduction of the feed, you have the growth of cells, which is being represented by the green curve, increasing all the way up. So that is a way to increase the growth phase. And once you increase the growth phase, the stationary phase will also be increased. Because once you keep, feeding the system, cells will be growing, cells will be producing. And so it means that you need to have a system that has larger space enough that within some intervals, when you introduce more feed, there is enough space for more growth, and there is enough space for more product that will be produced until maybe over a certain period of time. If you repeat this process, you would have increased the yield of your product. So that this is, as I said earlier, this is an improvement of the batch system, that you increase the yield this way by the introduction of feed into the system over some regular intervals over a certain period of time. And this is used in the production of also enzymes like galactosidase, which are used in the food industry. I think we saw an example of this, that galactosidase is used in some dairy products to make it wholesome for persons with some lactose intolerance, for instance. So that once you have that enzyme that breaks down lactose, then persons with some form of deficiency in that enzyme, can also consume these dairy products without any problem. Now, the very modern system of fermentation that is being used industrially is the continuous system. So, as the name suggests, the continuous system consists of pumps that are going to be continually pumping media into the reactive mixture, the processor or the digester, over the whole period of production. You have this continual feeding of the system so that you have a steady state condition. So you have a maximum production of your product over a long period of time. So this can be set up maybe even overnight, and the flow of media is set, a flow rate will be set very slow into the reactive mixture, so that what you see is that you have where you have chemostat vessel. That is where you are having the production occurring. And you see in that chemostat, you have an inlet of air. So that's oxygen has to be present. So that is one of the conditions for the microorganisms to grow and produce the products, the enzymes for us. And the feedstock is what you see in what is labeled as the media reservoir. And you can see that this media reservoir is connected to that chemostat vessel with a pump. So the media pump is what pumps the media from the media vessel to the chemostat vessel, which is the processor where the production is occurring. And so you see there again, you do see there is a rotor. So this is more like a laboratory. This is a laboratory setting, but it is this which will be, brought up, scaled up to an industrial level. If you look at it in an industry, you have the processor, actually a large vessel. That's the digester in which the production is occurring, and the media can just be a small bottle, the media reservoir, not so much of a big thing, and just have some. The pumps are also not any big pumps, little pumps that will be pumping this media into the digester slowly over a period of time. And once this media is present for the microorganisms that are in the chemostat vessel, you have the production ongoing. And so this will continue over a certain period of time. And once this is continuing, at the same time, up to a certain time, there can be harvesting of the product starting. So you see why it's continuous. There is that flow of media into the digester where the production is ongoing, and at the same time, there is a flow of the products into the harvesting vessel. So harvesting can be occurring at the same time as production is occurring. It's not like the batch system where when you have production occurring, you have to wait till the end before you can open the system now and then you harvest. Or like the fed batch system, that's the same way. Even though at intervals you introduce media, you still will wait till the end before you are able to harvest the product. In this case, you have the production ongoing, and at the same time, you can be harvesting over a period of time. So it gets to the very end of the production where you have all the media or flowing into the digester and is being used by the bacteria, and the product is produced up to the last end and is harvested, and it can be, and then is purified. So that is the continuous system that is highly efficient. It is actually the modern industrial system which is used in bioproduction or in this bioprocessing in industry at such large scale. That's what you see now in industries. Now, if you put all of this together, I mean, this is just to put all of it together for you to see the differences. So in the middle, you have the chemostat, what is the continuous system. And you see that, yes, the feed is connected directly to that, the processing system. And at the same time, you have the harvesting system also connected so that you have that flow of the process of feeding in, the media, production ongoing, and then harvesting ongoing at the same time. If you compare that to the batch system, you see the batch system on the left, everything is fed into the system closed, there is no separate media reservoir. There is no separate um, harvesting vessel also separately connected to it, but the whole system, the feed is in it already, and so it's just closed. Production goes on up to the end, and then the effluent valve is opened and the product is harvested. You compare that also to the fed batch. You notice that there is no uh, harvesting vessel which is connected to that um, production system, but you have a feed reservoir which is connected so that at intervals you have the introduction of the feed into the system. So you can see the differences between these three industrial fermentation systems all at once. All right. Now another microbiological technique which is being used in industries is what you refer to as genetic engineering. And here too we see that this is an example of, say, for instance, you have some microorganism, it's some bacteria, and you want this bacteria to produce a certain protein which you want to introduce into some food products. And this protein, for instance, if it's an enzyme, just like we introduce this enzyme into, say, dairy product, for instance, like the galactosidase that we were referring to much earlier. And the gene that codes for this protein, this enzyme, you can get it, say maybe from chicken, from cattle. I mean, and introduce it into the bacteria, which is the microfactory, which will be producing the protein for you. And so once you take that gene and introduce it into the microorganism, the bacteria begins to produce, because now the gene is in the bacteria and can get expressed. The protein can now be synthesized in the bacteria and be released. So once the protein is being produced and being released, that protein can now, be collected, so purified. So what it means is that once you have this, first of all, the media with the bacteria produced in, usually you produce it in a smaller quantity. So let's say you have that smaller quantity being produced, you introduce it into the big digester. All the systems are set so that you have an optimum condition, and over a certain period of time you have the bacteria producing lots of these proteins. And once you harvest, of course there is some waste. In this case, what you notice is that the product is being produced by the bacteria extracellular, like the product is being released out of the cell, so that the system is designed in such a way that at the end there is separation of the cells, I mean the bacterial cells, and even the if some of these bacterial cells are broken with their nucleic material, these can be separated as waste, and you have the product, the protein, which can undergo further purification before it is introduced into the food products, like say some dairy products, some beverages, like maybe if it is some amylases, pectinases that have been produced, these are then used for the production of these products. Just like we saw much earlier, that was in the last lecture, we saw that these enzymes were being used in the production of things like bread, cakes, beers, milk products like cheese. We are seeing it, here, being depicted that these enzymes are then being introduced into these food products. Now, we then look at what we refer to as a recombinant DNA technology, which looks very much like what we have just looked at, but not entirely the same. Because if you pay attention to what we just looked at, that was say a whole gene that was introduced into the bacteria, and then the bacteria was expressing that gene, was producing a protein from that particular gene, which was introduced into the bacteria. But in this case of recombinant DNA technology, what you see is that there is a gene that is coding for, say, insulin. So the hormone insulin, which is responsible for keeping blood glucose levels normal, and you want to have a gene, you want to have the production of insulin in large amounts. So of course, this will be an application in the pharmaceutical industry where you want to produce insulin and use it to produce some pharmaceutical products for the management of diabetic conditions. And you see that there is the gene that codes for the production of insulin. It's a protein. Insulin, that's a protein. And so you have a gene that codes for the synthesis of that. Now, that gene, with the use of some restriction enzymes, is obtained, let's say, from a human cell, and is then introduced into the plasmid, that is the nucleic material of bacterium. And so you see that the genetic material or the plasmid which is produced is now a recombined DNA, recombined nucleic acid. And so you see that you have the plasmid, the original plasmid is blue, and the gene that codes for insulin is incorporated into that plasmid, and that plasmid is then introduced into the bacteria, and the bacteria begins to express the gene, the insulin gene. So it produces a lot of insulin. So these the structures that you are seeing as VV, this orange, depicting that a lot of insulin is being produced by the bacteria. And these can then be separated, it's harvested, purified, and then it's used as an active ingredient to produce some pharmaceutical product for the management of diabetic conditions. So that is a difference between, even though you can say that recombinant DNA technologies is some form of gene engineering. You can say that's a subset of gene engineering, but this is specific because it refers to the combination, a recombination of the genetic material, of a bacteria that will result into the synthesis of specific products for us. Now, we do see also that you have systems of, say, yeast that are also engineered in this manner, to also, at such levels of, to produce such, levels of recombinant enzymes. And these enzymes usually are, say, invertase, glucose oxidases, proteases, And once they are produced, they get harvested, purified, and then they can be applied in, say, the food industry, because this enzyme specifically will be acting in the food industry. Of course, something like proteases, for instance, we did see that it can also be applied in different fields, like, say, maybe in the leather industry, the meat industry as tenderizers, and then even in detergents, for instance, we did see the application of this, for instance, for the removal of stains, like some protein-based stains, things like that. Apart from these yeast systems, you can have it like what we did see with uh, the the recombination DNA technology. What we did see there was, yes, particularly the use of bacteria, and usually common bacteria used is like E. coli. That you have the introduction of these genes, and then out of it you have the production of some particular enzymes. And so if you have just a breakdown, what you do refer to as cell lysis. So you break down this. Such cell lysis can be mechanical. I mean, in the industries, they have some uh, mechanical lysis of cells, and they do also have some chemical lysis. Then you once you have that, I mean, with processes of centrifugation, all of that, I think you did some of these things in the lab. You are able to have a separation of the broken down material of these cells and the product which you are looking for, which is say a supernatant, and then that can be separated, and you undergo further purification processes until you now have your your purified protein, which will be the enzyme which will then be used in other industries. I think I really will have to look for the links for these industrial processes, uh, so that you can watch these videos, and it will give you much more understanding of these things practically than just looking at it like this on slides and maybe opening books and be reading. Uh, it will not be so much interesting that way. All right. I think we'll take a very last one, and then that will be it. We've been talking too much for how many? More than 3 hours. We have, modern system of what's referred to as a CRISPR. Uh, it's one of these systems that these molecular biologists now so have been referring to so much, which is a more specific system of um, targeting some specific genes, and sort of is sort of reproducing them. Is you have some genes that have some repressors, so that when you have um, uh, you have a system where you can actually target these um, repressors of the genes, and once you are able to target this, it's a more precise form of targeting this the specific genes that you are interested in, that are responsible for the production of some enzymes like the lipases and cellulases, so that out of it you have some restriction at specific points. And once you have such restrictions, you can carry out things like um, um, how do you call it, these polymerase chain reactions and do reproduction of these genes, multiply them, and then you can have production of more and more of these enzymes, and then those enzymes can then be purified and become products which can be used in industries for the production. The applications that we saw last in the last lecture. You can see it in that. All right, I think this is where we will end it, and then if you have any questions, any contributions, if some of you are working in some industries, I mean, you should have a lot to share with us so that we can also learn from you. So the floor is open. If you have anything, you can write in the chat or you can raise your hand and speak. All right. So to my understanding, it means there is no evening student for SBT 458, Industrial Biochemistry. All the students are now weekend students. Is that the case? Confirmation? Mary, can you write in the chat? Okay. All right. So confirmed that for industrial biochemistry, all the students are weekend now, and so we have only weekend